Our group will be presenting on bioprinting and tissue engineering, a technology that combines biology and engineering to create artificial tissues for medical and research applications. We are Group 1. Our team members are Nur Aisyah binti Ayub, Shubaika binti Badrul, Amirul Faris bin Muhammad Sulong and Muhammad Hadi bin Abu Bakar.
Assalamualaikum warahmatullahi wabarakatuh. My name is Amirul Faris and I will be explaining on the introduction part and the mechanism part of our flipbook assignment. So first up is on the concept of sustainable tissue engineering, whereby tissue engineering restores damaged organs by using biology and engineering. This process is then automated through the use of 3D bioprinting, where it prints living cells layer by layer. And instead of using synthetic plastic, bio-based materials are used and are able to mimic the body and reduce medical waste. So then what goes into the printer? The material that goes into the printer are known as bioinks, where it is a type of hydrogel that protects living cells during printing. Some of the sustainable ingredients that use as bioinks are alginate or glycosan and also DECM, which is the decellularized matrix. Next up is that there are three main steps in the process of bioprinting, which is step 1, imaging, where a CT or MRI scan creates a digital model of the patient's defect. Step 2 is the printing. The printer reads the digital model and deposits bioinks in precise layers and building shape. And the last step, step 3, is the maturation, which the printed part goes into a bioreactor or incubator where the cells multiple and fuse into solid tissues. Next up is on the mechanics of the printing techniques. There are two deposition mechanics, which are extrusion based and also inkjet based. Extrusion based is done where continuous filaments of bioink pushes through a nozzle, and it is best for structural parts such as bones, but also creates shear stress. Inkjet based, on the other hand, are done through tiny droplets of cell sprayed onto a plate using thermal or acoustic energy. It is fast and precise but requires to have low viscosity bioinks. Lastly is on the cycle where interaction and biodegradation occurs. The cell material interacts and doesn't just sit there. The cells use receptors called integrins to grab the scaffold, and the stiffness of the biomaterial tells the cells what tissue they need to be. Biodegradation, on the other hand, is where the scaffolds are temporary. As new tissue forms, the bio-based material dissolves harmlessly via hydrolysis of enzymes.
Assalamualaikum, my name is Nur Aisyah binti Ayub and I will continue discuss in sustainable impact of bioprinting. First, bioprinting uses renewable and natural material. Most bioink are made from material like alginate from seaweed, gelatin from collagen, and cellulose from plant. This material can be replaced naturally and safer for the environment, which helped reduce the use of petroleum-based plastic and support sustainable resource use. Second, bioprinting uses low energy compared to traditional manufacturing. The printing process happens at a room temperature or a body temperature, so it does not need high heat or heavy machine. Because of this, less electricity is used, which helps reduce carbon emission and energy waste. Third, bioprinting helps reduce waste. The printer places the bioink only where it is needed, layer by layer. This means very little material is waste compared to cutting or modeling method. Some unused bioink can also be reused, making the process more sustainable. Fourth, most bioprinting tissue and scaffold are biodegradable. After implantation, the material slowly breaks down inside the body into safe substance. which this avoid long-term waste and remove the need for extra surgery to take out implants, making it more safe for patient and the environment. Last but not least, bioprinting can reduce animal testing. Bioprinted tissue can be used for a drug testing and disease study instead of using animal. This is more ethical and also reduce laboratory waste and environmental impact related to animal experiment.
My name is Baika binti Badru and metric number 230070, and now I would like to present about ethical and social value of bioprinting and tissue engineering. Bioprinting and tissue engineering are revolutionary technologies that allow scientists to create living tissue and organs. While these innovations offer great medical benefits, they also raise important ethical and social concerns that must be carefully considered. First, patient safety is the most important ethical concerns. Bioprinted tissues must be biocompatible, meaning they should integrate into the human body without causing immune reactions or harms. To protect patients, these tissues must undergo strict testing and evaluations before being used in humans. Ethical responsibility ensures that innovations that does not come at the cost of patient safety. Bioprinting also offers ethical advantages by reducing the need for animal testing. Laboratory-grown human tissue models can be used for drug testing and research, reducing animal sufferings and providing results that are more accurate for human physiology. Another major ethical issue is fair access to bioprinting technology. Because bioprinting is expensive, it may initially benefit only wealthy countries or communities. Ethical practice requires governments and institutions to support policies that ensure equal access and prevent a technology gap in healthcare. Bioprinting often uses human cells, including stem cells. Donors must give informed consent, meaning they fully understand how their cell will be used, stored, or possibly commercialized. Respecting donor autonomy and privacy is essential in ethical biomedical research. Bioprinting does not fit neatly into existing medical regulations, as it combines elements of drugs, devices, and biological materials. Clear regulatory frameworks are needed to ensure quality control, assign responsibility, and protect patient safety. Strong regulations help build public trust in this emerging technology. Ethical concerns also arise when bioprinting goes beyond therapy and moves toward enhancements. The creations of designer organs could affect human identity and social equality. Many experts argue that bioprinting should focus on medical treatments rather than enhancements to avoid ethical controversy.
Assalamualaikum, my name is Muhammad Hadi and we will moving on into Sustainable Development Goals number 3, which focus on good health and well-being. Bioprinting and tissue engineering strongly support these goals by enabling the regeneration of damaged or diseased tissue using bioprinted scaffold. These scaffolds are designed to be highly biocompatible, which help reduce immune rejection when implanted into body. This improves patient safety and increase the success rate of treatment. In addition, many bioprinted implants are biodegradable, meaning they are naturally break down as new tissue form. This reduces the need of additional removal surgery, lowering medical risk and improve recovery outcome. Overall, sustainable bioprinting contribute to safer, more effective and patient-centered healthcare solution aligned directly with SDG 3. Next is Sustainable Development Goal 9, which emphasizes industry innovation and infrastructure. Bioprinting represents a major technology advancement by integrating 3D imaging, automation, and advanced biomaterial into medical manufacturing. This technology drives innovation in the biomedical industry by enabling the production of customized tissue and implants with high precision. Compared to conventional manufacturing, bioprinting uses low energy and high control process, making it more efficient and sustainable. Furthermore, the development of bioprinting supports the growth of modern and sustainable medical infrastructure, especially in regenerative medicine and personalized healthcare. Therefore, bioprinting not only advances healthcare outcomes, but also strengthens innovation-driven and sustainable industry development under SDG 9. Lastly, we focus on responsible consumption and production, which is SDG 12. Bioprinting directly support these goals through its efficient use of material and environmentally friendly design. The technology uses renewable and bio-based materials such as alginate, chitosan, and decellularized extracellular matrix as bioin. These materials are biodegradable and reduce dependable on petroleum-based synthesis plastic. Additionally, the layer-by-layer printing approach that ensure only the requirement amount of material is used, minimize waste during production, and any unused bioin can often be reused, further reducing resource consumption. By producing biodegradable scaffold, bioprinting also lower long-term medical waste, making it more responsible and sustainable approach to medical production. In conclusion, sustainability plays a crucial role in future development of cell tissue and engineering, especially with the use of 3D bioprinting technology, use of renewable and bio-based material, support the tissue regenerative while minimize environmental impact, and these materials are both degradable and biocompatible, allow the scaffold to support cell growth and safely degrade as natural tissue form. Moreover, bioprinting improve efficiency through precise layer-by-layer fabrication and low-energy heat process. Significantly reduce material waste when combined with ethical considerations such as patient safety, reduced animal testing, and equitable access to treatment. Sustainable bioprinting ensure responsible medical innovation, and overall, sustainable tissue engineering represent the balance between technology advancement, ethical responsible, and environment protection, shaping more responsible future for healthcare.